Today we're going to finish up talking about genetics, guys, and we have two more types of crosses to go, those involving multiple alleles and those involving sex-linked traits. And we're going to start with multiple alleles. Multi means many, and alleles are forms of a gene. So multiple alleles have many different forms to that trait. And so far, everything we've talked about only has two forms. You either have freckles or you don't. You can roll your tongue or you can't. You have a red or a white flower. And even with incomplete and codominance, we could get a third phenotype, but that third phenotype is a mixture of the two alleles for that trait. So the only type of cross you need to be able to do with multiple alleles is blood typing. And blood types have four different forms. You can have A, B, AB, or O blood. And there are three alleles for those blood types, A, B, and O. Now remember, on the outside of a cell, you have these proteins that mark the cell for what it is and allows it to communicate with other cells. So in A blood, you only have the A protein. That's it, okay? With B blood, you only have the B form of that protein on the outside of the cell. But A and B are codominant. And remember, in codominance, you show both traits separately and equally. There's not a blending of A and B here. We have B proteins and we have A proteins on the outside of that cell. With O blood, you have no protein. There's nothing there, okay, to trigger any sort of response. Keep in mind that to get any genotype, you have to have two alleles. You have one from mom and one from dad. So you can't carry all three of these at one time. You can only have two. So with A blood, you can have a homozygous person for A, so they would have two A alleles. You could have a heterozygous person for A, and they could have an A allele and an O allele, because O is recessive. It's going to hide behind A or B. For B blood, you could have a homozygous person for B, and they would have two B alleles. Or you could have a heterozygous person for B, and they would have a B allele and an O allele. AB blood, you have to have an A and you have to have a B allele. And to get O blood, remember, O is recessive, so the only way to get that recessive trait is to have two copies of the recessive trait. So you would need to have O blood. So when it comes to blood typing, you have to make sure that if you're going to give someone blood, that they have the right blood type being given to them. So a person with A blood has these antigens in their body. These are the antigens, and they recognize this A blood. You also have antibodies that are going to recognize the B blood and fight against it. Okay, anti means against. So B-blooded people have the B antigen to recognize this B protein, but then they have this A antibody that's going to recognize and fight against the A protein. People with O blood have both antibodies. They're going to recognize A and B blood and fight against it. And people with AB blood do not have any of these antibodies. So they're going to accept blood pretty easily. So when it comes to an emergency and you need to give someone blood, if you give them the wrong blood type, these antibodies are going to fight against that blood and you'll end up killing this person. So it's really important that you give someone the correct blood type in an emergency situation. So people with A blood can take other A blood, or they can take O blood because it has no protein that's going to be picked up by these antibodies. So you can get A or O here. People with B blood can have other B blood, or they can also take O blood. Again, O has no protein to interact with these antibodies or antigens. With AB blood, a person can recognize A blood because they have these A antigens. They can recognize B blood because they have these B antigens. And O blood has no protein. So a person with AB blood can take A blood, B blood, AB blood, or O blood. They can have anything. And AB positive is considered to be the universal recipient. We're not going to get into positives and negatives. This is just a different type of protein. O blood has no proteins present on it, and it will pick up any other kind of blood that's in the body. So the only type of blood you can give an O person is other O blood, because they're going to recognize these proteins and these proteins, and they will fight against those. So when it comes to positives and negatives, this is just a different type of protein. Positive people can take positive blood or negative blood, but negative people can only have negative blood. They will pick up that positive protein if it comes in their system, and they will fight against it. So O O negative is considered to be the universal donor. AB positive is considered to be the universal recipient. They can take any kind of blood, and O negative can give to anybody because they do not have any proteins that would trigger an immune response in anyone with these other blood types. So let's do some practice here. Okay, so we have a baby that has type O blood. So we know that its genotype has to be OO. We have a mother who has A blood, and we don't know if that A blood is AA or AO. It has to be one of the two. And we have a dad, and he has B blood, and we don't know if his genotype is homozygous for B or heterozygous, and he has BO. So since the baby has O blood, we're going to go with the parent that has the best chance of giving the child this O blood to see if he could possibly be the father of the baby. Well, we know if the baby has O blood, that this genotype would not work. There is no O allele there for mom to donate. So we're going to use the heterozygous form of A blood. And we are going to go with this one because we actually have a chance to pass on that O allele. So if we cross these two parents in the same manner that we have been crossing, then we will find that you can have any blood type from this cross. You can have AB, B, A, or O. And since you have a 25% chance of having that O blood, we can't say that he's not the father. There's definitely a possibility that he could be. Now we're going to look at your second practice problem, and we have a baby that has been switched in the hospital, and the baby has A blood. So we know that the baby's blood type is either homozygous for A or heterozygous, and it has AO. We know that couple one has AB blood and O blood, so we know their genotypes. One parent is AB, and the other one is OO. When it comes to couple two, they're a little trickier. They have O and B blood. So we know the first parent has OO, and the second parent is either homozygous for B, or they are heterozygous for B, and they would have BO. So we don't know which one to use, and we're going to use the one that gives us the best chance. So this baby's been switched in the hospital. Let's see if we can figure out who the parents are just by their blood type alone. So couple one, we'll go with first, they have A, B, and they have O blood. And so when we cross these two, you're going to find that you have A blood here, 50% chance, and you also have a 50% chance of getting B blood. Remember, all it takes is one allele that's dominant here to have that type of blood. So we do have a chance of getting this child right here. Now we need to do the second set of parents just to see if we could get A blood with them. So we have OO, and we have, let's try BO first. And we can see when we have BO mixed with OO, you have a 50% chance of getting B blood and a 50% chance of getting O blood. So no way to get this baby with that genotype. And we will try BB as well just to make sure we can't get that genotype. So we'll replace that O with a B. And now all of the children have B blood. So there is no way that this couple could be the parents to this kid. So we know that it has to be couple one. And you may notice that there is no way to get this baby from this parent because there are absolutely no A alleles here to donate to this child. So now we're going to do the last cross that you have. We have a baby with AB blood, so we know its genotype has to be AB. We have a mom that has A blood, so we know she is either homozygous for A or she is heterozygous for A. And we have a dad who has O blood, so we know for sure his genotype. He has to be OO. So when we do our Punnett square here to figure out if he is the father, we're going to put mom's alleles and dad's alleles on our Punnett square. We'll start with dad's. We know his genotype. And we'll put mom's in here as well. Now, to have this baby, you may notice there are no B alleles, so you're probably already figuring out that, no, he cannot be the father. But when we do our Punnett square, you can see for sure. There are no B alleles to donate, no way to get this AB blood if this is the dad. Um, we can also try the A allele for mom. Oops. And you'll notice that when you try the A allele, the homozygous genotype, there's still no alleles for B and no way to get this child. So he is not the father of this child. No need to do an expensive paternity test there. You can tell by simple blood typing. Now, keep in mind that you can't always tell if a child is belongs to a certain dad or a certain set of parents based on blood type alone. You would have to do some more intensive genetic testing, but this is a good place to start. So let's just review your possible blood types and their genotypes. So you could have A blood, and that's either going to be homozygous for A or heterozygous for A. You could have B blood, and you could be homozygous for B, or you could be heterozygous for B. With AB blood, you have to have an A and a B allele. And with O blood, you have to have two O alleles. The only way you're going to get that recessive genotype. And just to show how to do your geno and phenotypic ratios, let's cross a heterozygous person for A with a heterozygous person for B. And you can pause the video and try this yourself if you want. And now that I've got my Punnett square set up, if I do the cross, I've got an AB person here, a heterozygous for B here, an A person here, and an O person here. So my ratio, my genotypic and my phenotypic ratio would be 1 to 1 to 1 to 1. For every one of these, I got one of these or one of these or one of these. If I wanted to do a geno or phenotypic ratio on a different blood type, let's say we were crossing AB with BO, then now things are a little bit, now if I look at my geno and phenotypic ratio, things are a little bit different. I have a genotypic ratio of 1 to 1 to 1 to 1. All of these are different genotypes, but my phenotypic ratio is 1 to 1 to 2. I have one person with AB blood, one person with A blood, and I have two people with B blood. So geno and phenotypic ratios can be different here when you're doing blood type. So now we're going to look at sex-linked traits. Mammals have two types of chromosomes. You have autosomes, and you've got 22 pairs of autosomes, so for a total of 44. And you have two sex chromosomes, or one pair of sex chromosomes. And your sex chromosomes come in two different types. You can have X's or you can have Y's. And to get a girl, you have to have an XX genotype. To have a boy, you have an XY genotype. You can think about the Y as being dominant. It's going to dominate over these female characteristics, and you're going to end up with a male. So sex-linked traits are determined by what's on these chromosomes, these X's and these Y's, and they're always going to be attached to these sex chromosomes. Most are going to be recessive, and most are carried on the X. The X is much, much bigger. You can see an example of that here. Here's an X chromosome. Here's a Y chromosome. So the X has more space to carry more genes, kind of like a suitcase. The bigger the suitcase, the more genes it can carry. But all right. So anyway, whoops, didn't mean to do that. Most of these sex-linked traits are going to be recessive, and that means you're going to need two copies of this trait. And these are some examples. Hemophilia, which is a bleeding disease. You can actually die from a paper cut if you don't get treatment and you have hemophilia because your blood can't clot. So it's going to continue to bleed and bleed and bleed. Baldness, male pattern baldness, going bald as you get older, that is a sex-linked trait, and it's carried on the X chromosome. And then albinism is sometimes carried on the X chromosome. If we look at the effects of sex-linked traits, you'll see that males are more often affected than females. Most of these genes are carried on the X chromosomes, and boys only have one of those. And so since they only have one, they're going to show whatever is on that X chromosome, whether it's dominant or recessive, it doesn't matter. They only have one trait for it. So if we think about color blindness is an X-linked recessive trait. And 1 in 12 men worldwide are colorblind, whereas 1 in every 200 women are colorblind worldwide. So we're going to do some practice with this. And before we do, we are going to look at how you would write some of these genotypes. So remember, in order to have a girl, you need two X chromosomes. And in order to have a boy, you need an X and a Y chromosome. Now, when it comes to expressing traits on these chromosomes, you're only going to attach to the X because the X is what's carrying the gene. So if I want a person who has hemophilia, then I'm going to use a little h because remember, hemophilia is recessive. And if a person does not have hemophilia, they're going to have the normal form of the trait, which is dominant. Okay. So if we look at girls, in order for a girl to have hemophilia, she will need two copies of the gene for hemophilia. She's got two X chromosomes. In order for a boy to have hemophilia, he only has one X chromosome, and on that X, he's going to have the trait for hemophilia. We never attach anything to the Ys. So for a girl to carry hemophilia, she would be heterozygous. On one X, she's going to have the normal copy of the trait. And on the other X, she will have the recessive hemophilia version of the trait. So this means she carries it. She doesn't have it because she still has that one dominant trait to cover it up. In order for her to be completely normal and have no traits for hemophilia, then both X's will have the dominant form of the trait. And it works that way with all of your sex-linked genes. So for a boy to have hemophilia or to not have hemophilia, he would have to have the normal version of the gene on his X chromosome. So this person has hemophilia, this person does not, this person does not, this person does, and this person does not. Okay? So keep that in mind when you are making your genotypes. You still have to have these sex chromosomes with sex-linked traits, and you'll have to attach something to those X's. Keep in mind, not all traits are sex-linked traits. Very few are. The X chromosome is only so big. So now that you know this, please do not try to make every trait a sex-linked trait because they're not. And some of you will try to do that, but please keep in mind, you will have to be told if the trait is sex-linked. And if you are told it's sex-linked, that's when you would use X's and Y's. Under no other circumstance would you. Blood type, for example, not sex-linked. We don't use X's and Y's for that. So now we're going to do some practice problems with sex-linked traits, and these are the only two we're going to do. Keep in mind that you will need to use X's and Y's because you've been told these are sex-linked. So the first step to solving your genetics problems, determine the type of cross, and we've done that. The next step is determine your genotypes of your parents. So we'll need to do that with both of these problems. We'll need to put those genotypes on a Punnett square and do the cross. That's the third step. The fourth step is analyze the Punnett square, and the fifth step will answer the question. So we have a woman carrier, and her father of her child is a normal male. So we need to figure out what are the chances they will have a hemophiliac baby, and what is the probability that it will be a girl. So we know this is a sex-linked cross. Let's go ahead and make the genotypes of our parents. So we have a woman who is a carrier. She is a girl, so she has X, X. And since she is a carrier, she has one normal gene for hemophilia, and she has one recessive gene for hemophilia. The man has XY. We know that automatically. We attach nothing to the Y. And since he is a normal male, he will have a normal copy of the gene. So now we've got our genotypes of our parents. So we're going to put that in the Punnett square. And it doesn't matter where you put the girl or where you put the guy, as long as you don't cut your corners and you keep their alleles on the same side of the Punnett square. So I got my girl there, put my boy here, and now I'm going to do the cross. And you do have to include these exponents. So I've got my X big H and my X big H. Now we need to analyze our Punnett square. So we're going to go through and figure out what genotypes we have. And remember, you have to include the gender here. So I've got a girl, and she does not have hemophilia. I know that because she has at least one big H. Here I have another girl, and she does not have hemophilia. She has a big H. Here I have a boy, and he does not have hemophilia because he has the big H. And here I also have a boy, but he does have hemophilia because he has the one copy of the gene. So in order to answer the question, what are the chances they will have a hemophiliac baby? 25%. And there's the 25% right there. What are the chances it will be a girl? Zero. Both girls do not have um, hemophilia. If I wanted to find the geno and phenotypic ratio for this, well, I've got two girls. Let's see, let's do the genotype first. So I have one girl that is homozygous dominant. I have one girl who is heterozygous. I have one boy who has the trait for hemophilia, and I have one boy who does not. So there's the one that has, and there's the one who doesn't. So I've got a genotypic ratio of 1 to 1 to 1 to 1. If I look at my phenotypic ratio, I have a girl without it and another girl without it. So I've got a 2, and I've got a boy without it and a boy with it. So my phenotypic ratio would be 2 to 1 to 1. Just remember, when doing these geno and phenotypic ratios, you have to look at whether or not they're a girl or a boy, in addition to whether or not they have the trait. So let's look at the next practice problem. So we have a man who is colorblind, and he is having a child with a woman who is heterozygous. She is a carrier for color blindness. We need to know the chances that they will have a colorblind kid and the chances that that kid will be female. So remember, we need to determine the type of cross. We've already done that. We know it's sex-linked. Now we need to determine our genotypes of our parents. So we have a girl, and she is heterozygous for color blindness. So we are going to give her a big B and a little b. And remember, color blindness is recessive. So in order to be colorblind, she would need 2 little b's. We also have a guy, and he is colorblind. So he will have the recessive form of that trait. We are going to do our genetic cross. So now we have a man who is colorblind, and he's having a child with a woman who is heterozygous. So we know the type of cross. It is a sex-linked trait. Color blindness is recessive, so we're going to use little b's to express color blindness. Now we need to figure out the genotypes of our parents. So we have a guy, and he is colorblind, so we're going to give him a little b. And we have a woman who is a carrier for color blindness, so we're also going to give her a big B and a little b. And now we need to put these parents on this Punnett square and fill it out for step 3. And when I do that, okay, so now that I have my Punnett square filled out, I need to go back and analyze each person. So I know that this is a girl. She has 2 X chromosomes, and she does not have color blindness. And I know that because she has a big B there. I have a boy here because he has an X and a Y, and he does not have color blindness because he has a big B. He has the normal version of the gene. Here, I have a girl. Whoops. She does have color blindness. She has both recessive forms of the trait, and so she has no choice but to show that. And here I have a boy, and he does have color blindness because he has the recessive form of the trait and no chance to cover that up because he doesn't have a second X chromosome. So if I wanted to do my geno and phenotypic ratio here, my genotypic ratio is 1 to 1 to 1 to 1. All of these are different. X big B, X little b, X little, X big Y, X little y. So all of those are different. If I want to look at my phenotypic ratio this time, I have a girl who doesn't have it, a boy who doesn't have it, a girl who does, and a boy who does. So I've got a 1 to 1 to 1 to 1. They are all different as well. If we look back at these questions to answer them, what is the probability they'll have a colorblind child? Well, 50%. There was a girl and a boy who had color blindness out of those 4. And what is the probability that the child will be colorblind and female? Well, 25%. We only had one girl who had color blindness out of those 4. If the question asks you, out of the colorblind children, what are the chances it would be a female? 50%, because there were 2 colorblind children and only one of them was female. Polygenic traits are the last thing we have to talk about. Poly means many, and for genic, well, think genes. So these are traits that are controlled by 2 or more genes. And that means they have more than one place that they are coded for in your chromosome. So you end up with more than 2 alleles for these traits. Mom and dad both give you 3 alleles for your eye color. I think it's 3. And so you end up with many different forms of eye color. So you end up with 6 total alleles for eye color. Hair, I think you end up with 12 alleles. Each parent gives you 6, and those hair color genes are different places in your genome or in your chromosomes. So polygenic traits are things you often see gradients of. Other examples would be height. You are not just 7 feet tall or 4 feet tall. There's everything in between and above and below that as well. Skin color, these are an example of all the different types of skin color that you could have. And then we already looked at eye color. So polygenic traits coded for in many places. There's many different types of those traits. Just one fun one before we go. Coat color in cats is sex-linked. It's found on the X chromosome, and coat colors in cats are codominant, at least black and orange. So you might have had a calico cat before and noticed that they're all females. So let's say that we have a black female cat. In order for her to be black, she has to have both black alleles. And we have an orange male. So in order for him to be orange, he has to have the orange allele. And we're going to put those together. So when you mate a cat that is female and black to a cat that is male and orange, you have a 50% chance of getting a calico cat, and you have a 50% chance of getting a black cat. Now you might notice that the only way to get a calico cat is to have a girl, because you have to have two X chromosomes and they each have to carry a different allele. You can have a male cat that is calicoed. It's really rare. You would have to have two X's each carrying a different coat color, and then you would have to have this Y. And there's usually something that's going to be wrong with these cats, because you really should not have 3 chromosomes here. So these cats are often abnormal. So that's it with genetics. I hope that you learned something. I hope that you had fun doing these crosses. It's pretty cool to know a little bit about how you can breed things together and get different traits. If you have any questions, please contact me. Other than that, make sure you've done all your practice, and then try your worksheets and online Canvas quiz, and then you have a quiz in SchoolNet after that. Hope to hear from you. Thank you.